This is excellent. I want to thank everyone for joining today. We are thrilled to be sharing some what we think are quite interesting results from a novel study done in conjunction with Dalhousie University and Dr. Carmen Giacomantonio. We're going to move quickly into the data associated with the first phase of the study that have these interesting insights. But first, we do want to set some brief context for you to better understand where we're coming from and what we're doing in this study aiming to achieve. First and foremost, Sona Nanotech is developing a photothermal therapy called targeted therapy, which involves the intratumoral administration of gold nanorods into solid tumors and the subsequent exposure to near-infrared light, gently heating a tumor from the inside out, selectively killing the cancer cells. However, with the study data we're sharing today, we're seeing something that's much more interesting that we're keen to dig into with you. So here we go. So, of course, today we'll make a number of forward-looking statements, and as a publicly traded company, we refer you to our disclosure documents and the risk to future unknowns identified in them. Sona Nanotech does one thing and does one thing well. We make gold nanorods, and they're the only gold nanorods that are truly biocompatible and don't involve the use of toxins. Now, lots of people make gold nanorods, lots of people make gold particles. Our gold nanorods, of course, as with any, are highly functional. So you can tune the aspect ratio so that they respond to different wavelengths. And gold nanorods have been shown to be the most efficient at thermal conversion of near-infrared light into heat. That's why we use them. That's what we have that's special. We're using them to develop as I say, photothermal therapy, and keen to show you those results. A last point here is that these materials, don't take our word for it, have been vetted by the Nanotechnology Characterization Lab. It was established by the FDA and the National Cancer Institute to accelerate progress of nanomedicine by providing developers such as ourselves with advanced testing and characterization of nanoparticles. And they have found through now 7 batches of material to contain neither endotoxins nor microbial contamination. We believe that this suits us ideally for in vivo applications such as photothermal therapy. Now, Immuno-Oncology, the largest drug in the world. It has a brand name, Keytruda, of course, did sales of 17 million 2001. I believe last year, 2023, with 25 billion. There's a significant amount of money being paid for immunotherapy drugs. It's a significant market, and that's because, of course, it's been shown that these immunotherapies have vastly improved survival rates versus cancer alone. This has been happening over the last 15 years. Everyone's sure aware of this. This was a study that was, in fact, commissioned by Merck, but compelling data nonetheless. So when they work extremely well and lift the game. However, response rates, as we see here, are still very limited across the board, depending on the type of cancer. And therein lies the rub, if you will. Which begs the question, why doesn't immunotherapy work more frequently? And to delve into this, I'm going to introduce and invite Dr. Carmen Giacomantonio, who's our principal investigator and a scientific advisor to the company, to discuss and tell us about this. Carmen.
Thanks, David. So there's a couple of reasons that you saw this slide, and I know most of you that are on know how effective immunotherapy has been at this point. Immunotherapy has been largely modulated through checkpoint inhibition, which means the immune system engages a tumor. As we know, most tumors are engaged, or many are, and then the engagement is enabled by taking or disabling the checkpoints that stop the immune system from progressing through to killing the tumor. When this fails, what's typically done today in medicine is that the checkpoints are inhibited. So Keytruda, pembrolizumab, the PD-1 inhibitors being dominant in the clinics. But when that doesn't work, we add a second checkpoint and a third checkpoint inhibitor. And the consequence, of course, is sometimes we get better immunity and we clear tumor, and sometimes we generate more autoimmunity and create disease or morbidity. And the reason is because mostly in those situations, the antigen that's been expressed, and this is from purely an immunological perspective, the weaker the antigen, the more enabling the immune system needs to keep focused on it. And so that's essentially what the research that I've been doing with Sona, our collaboration, is focused on. Next slide. So we know that there's lots of antigens in tumors. So, and we talk about antigen load or mutational load in cancers as distinguishing the cold from the warm tumors, and that's fantastic. What we do know is that even a cold tumor can be seen by an immune system if the right antigen is exposed and presented. And essentially, that's what the notion of the hyperthermia is to generate this situation in a tumor that when gold nanorods are placed into a tumor there's a number of modalities we can talk about, but when the tumor is filled with gold nanorods, infrared or near infrared light passing through the nanorods will excite them, create heat, and the heat will then go on to generate an apoptotic cell death. The heat that, next slide, the temperature range that we're looking for is in that 42 to 49 degrees, in the sweet spot in around the 45-degree range. And the reason that happens is because tumor cells are sensitive to heat, essentially. And so because of heat shock protein, because of apoptotic pathways that are triggered through the stress of heat, the tumor cell goes into an apoptotic death. An apoptotic death, unlike necrotic death and other forms of ablation, preserves protein. And so, and we know that when a tumor, or when anything in the body actually dies, that the immune cleanup is through the innate system, and that results in protein and therefore antigen presentation to the system. If the antigen is novel and robust, it will establish new immunity. So this is essentially the principle that we're exploiting. Next slide. So what we did, we saw the potential in the gold nanoparticle-based hyperthermia to expose or present new antigen to the immune system. And then we thought that in and of itself would be fascinating. And can we prove that this is in fact an immune phenomenon and not just an apoptotic to its endpoint pathway? And we asked the question, can we use standard clinical immunity today that we use in the clinic, that I use frequently every week, to enhance this process? So we chose an industry standard model. The models are primarily mirroring most models that we look for immunology and immunological understanding. And the model we started with was a 4T1 triple negative breast cancer model. Next slide. So essentially we took the model, we set up tumors in two flanks, in the right and left flank, or the left and right flank of the mouse. And we used this principle that we were going to heat the tumors, we were going to cause an apoptotic cell death, and that apoptotic cell death is going to result in antigen presentation that we are going to try to, we were bold enough to say, synergize using our standard immunotherapies. Next slide. And so the next slide, which is, we can just move right through that. We started with breast cancer because that's an industry standard, lots of familiarity with it. We are also looking in melanoma as the next phase of our research, and rectal cancer. All three of these tumors, as you know, have been successfully treated in the clinic to some one degree or another with immunotherapy. All of them have limitations because of the nature of the tumor, the location of the tumor, and the engagement of the immune system. And so they all represent a major impact and burden on the healthcare system and patient lives. Next slide. So this is where I'm showing you the first slide, which is this works. So this slide is essentially showing that the hyperthermia plus interleukin-2, a standard immunotherapy that is given currently in most centers in North America intralesionally, essentially flattens the growth curve in these tumors. If you look at the red curve, you're going to see that there's the hyperthermia once applied, and we were very excited. That's one application of laser to the gold nanoparticles that have been injected into the tumor, and we saw an 80% reduction in tumor volume. What we see, however, is that over time, the remainder of the tumor cells, the viable cells, will continue to grow to a point. The blue is the control, that's just PBS, and the gold is the interleukin-2. And the interleukin-2 line starts to flatten, but what's happening in these tumors is that they're growing beyond the tolerability of the mouse model. And so a lot of the mice at that point have to be sacrificed, not because of tumor death per se, but because of morbidity related to it. And next slide. So what we see here, and what's even more exciting, and this was really well beyond what I expected to see from this series of experiments, was that there is statistical significance. If you look at the effect of the gold nanoparticle, the hyperthermia plus the interleukin-2, these are statistically significant across the board. The last one, there's an up twist in the curve, and this in our data set is related essentially to one mouse that outgrew. Now that tells us that there's an opportunity. We gave one gold nanoparticle injection, one laser activation, and then we serially treat it with interleukin-2. That late uptick might be an indication that there needs to be a further treatment. When I treat melanoma in the clinics today, we know that in the responders, sometimes we have to treat for a number of a series of treatments to affect the complete change, and that may be related to the antigen and that engagement in that particular animal or person. Next slide. So in this slide, what we're seeing is that this is the why we kept this in here just to show you. There is the one outlier that if it wasn't there, that standard deviation would have gone pretty flat to look just pretty much like all the other ones. But, full disclosure, there was one that outgrew, and we could have a good discussion about what that actually represents. What it represents, of course, is that we have more to do to understand exactly what's going on. But that data shows clear and inarguable response with this combination. That's extremely exciting. Next slide. So the other thing that's exciting to me and to most of the people that are treating, so this is intralesional direct tumor injection. This is not systemic. However, what we're experiencing in these most, like as I explained early on, we do a two-flank model. Left flank is the treatment, the right flank is the observation. The treatment flank, of course, went flat. This is the contralateral flank tumor. And what we're seeing there is that we're having a contralateral response, indicating that this is a systemic response of immunity. And I'll show you why I'm saying it's immunity. And in the vernacular of the discipline, that's an abscopal response. That's the response that 10 years ago people would call the unicorn. That when you treat a single tumor and other tumors disappear. And so this to me was as exciting as the primary treated tumor going flat. And this is the immunity. And so this is CD4, CD27 positive. So these are T cells. And when you look at the percentage of T cells that we see upregulated in these tumors, we see that there's CD4, these are helper cells. Interesting, if you look on that slide, the PD-1 is expressed, which of course, all those of us in the immune world would say, well, there's a target that we can enhance the immune response, perhaps by interfering with those PD-1 expressing cells. We see lots of CD8. We see, if you look at the CD8 positive PD-1 cells, there's a statistically significant influx into those tumors. With looking at to the far right with the controls versus the gold nanoparticle plus IL-2. Similarly, we see something, if you look in the lower panel, the second from the left, we see memory cells. CD8 memory cells are important because that's an immune response that helps us to explain what's happening in the contralateral tumor. The same with the CD8 effector cells. The NK cells were, to my surprise, a little bit not as robust as we had expected to see in the tumor. And we see a B cell response, and my lab has particular interest in B cells and the B cell influence on tumor response. And we know that a B cell response in people can be associated with actually a diminished overall systemic response. So we want to monitor this and see what we can do with B cells as part of our research going forward. Next slide. So the next slide is to look at the lymph nodes. So Fox on the far left is FOXP3, CD4. So those are the T regulatory cells. Of course, we see T regulatory cell upregulation. We're going to see that in all immune responses. It might be related partly to IL-2 as well. So that's good learnings. But again, IL-2 is definitely causing tremendous effect in this. We see a tremendous effect in the clinic. The other things we see in the lymph nodes that this is a robust lymph node based immune response. This is all. So this is the left, which is the treated lymph node. We see lots of effector cells. We see lots of CD8 effector cells, and we see in the lymph node a significant uptick in the natural killer cell population, which of course is in response to that innate or that new antigen that we believe having all of this. Next slide. So I'm going to, Len, if you want to go on and talk about this is really the technical side of what we believe the gold nanoparticles are doing.
Well, sure. Thanks, Carmen. So as David mentioned earlier, what we're doing is photothermal therapy, and Carmen mentioned as well, converting light into heat. So gold nanoparticles are injected. Now they're truly nano. That's an image of a red blood cell. The little specks are nanoparticles in the red blood cell. They're truly nano, and they diffuse throughout the system. In the case of the current study, it's an intratumoral injection, not a systemic injection. In the middle, we see a melanoma patient, and this is actually one of Dr. Giacomantonio's patients who clearly has unresectable melanoma. It's locally advanced melanoma, so it's a prime candidate for an early first-in-human type study where the tumors would be injected, infrared light would be shown on the area, and then essentially you can deliver this 1-2 punch where you cause shrinking of the tumor, and then the apoptotic cell death releases the neoantigens, stimulating the immune system. In terms of target indication, did you want to address this, Carmen?
Yeah, so melanoma is this patient population that I see frequently are the melanoma patients, the stage 3B, 2C, and 3. These are people that have in-transit lesions, like the in-transit lesions that are on that person's neck. We see them in the trunk, we see them on the extremities. And these are often beyond what's felt to be resectable, or at least resectable without considerable morbidity. And of course, the stage 4, so the stage 4 M1, meaning that these are predominantly cutaneous mets, they actually behave well all in terms of prognosis if you can treat them. But as many of you will know, the cutaneous disease doesn't respond as effectively to the systemic checkpoint inhibitors. And so this lends itself to this kind of combination therapy. So the people that would be in target would be that class of patients who have failed immunotherapy, discontinued the immunotherapy because of toxicity, which of course is a significant consideration and experience, especially in the dual checkpoint inhibited patient. And patients that respond, but respond incompletely. So we see frequently patients who have systemic response, but then have progression in the accessible cutaneous mets. And so even in the clinic today, we are attempting to do things with intralesional interleukin-2. And I'm quite confident and excited that this would enhance that response as well. So we hope we have this collection of people that, this is if you look in the algorithm, the NCCN guidelines would say when you have these people with unresectable disease with local you have a myriad of things that you can do, but there's really no next step. And when you see the NCCN saying essentially best practice, this is tremendous opportunity to do not only best practice, but improve the response rate through some adjunct like the gold nanoparticle with an immune adjunct.
Thanks, Carmen. Do you want to advance that slide, Len? Excellent. So as we move this therapy towards clinic, back one for me. And we've been guided by numerous specialist consultants, including as a scientific advisor, Dr. Giacomantonio, but also large regulatorily counsel firms, and importantly, 2 different organizations. The second you'll know, but the first, Excite International, is an organization that partners small developers with novel cancer applications to leading physicians across North America and who work with both hospitals, universities, and importantly, payer organizations. We've had 3 panel discussions, and they have been instrumental in guiding us towards those indications for use and giving us counsel as to the most efficient path to get to the most powerful application for our technology. For the FDA, we've had one pre-sub meeting with them and the associated correspondence back and forth. That, of course, has been highly helpful and given us clarity on our proposed preclinical development plan. And we'll be going back to do a second pre-sub with them moving forward. But net a lot of support across the board from those 2 organizations, I'd say. I'd also say, oh yes, we were very pleased to have in our FDA discussions the NCL we mentioned off the top, the Nanotechnology Characterization Lab. Their personnel had offered and in fact joined us to sit with us at the FDA's meetings. And we've had one significant US payer from that Excite International panel who has offered to sit with us on our side of the table at the next FDA meeting. Obviously, payer organizations are really significant constituents, and they've been extremely helpful in helping us get the of data that from those organizations specifically, as opposed to the FDA. So, looking at what we have here, a number of these things, a number of the elements of our therapy have already been done. So we know heat kills cancer. We know that photothermal treatment using infrared light devices works to use those to heat up gold nanorods. Thirdly, there's precedent for the FDA approving nanoparticles injected into humans that was done for several prior clinical trials. And we've seen our targeted hyperthermia, THT, efficacy in small animals demonstrated in peer journals, as well as Dr. Giacomontoni's study of this data that we see now. Now, that's just the first phase, of course, of the study that's undergoing, and we'll have other ones coming soon to report back on. So looking forward to getting towards the clinic, we've got a we've been guided by physicians throughout North America, part of our input panel. We've had our first FDA application. We've selected our gold nanorod GMP, good manufacturing partner. Our efficacy study is underway. And then we are all set in number 6 and number 7 to initiate the safety, feasibility, and biocompatibility studies and initiate the regulator required GMP manufacture. Those things are all set to go, all leading to the goal of being first in human, getting first in human for this therapy in the next 12 months. We're very excited about that because we think it can make a great deal of difference, particularly when you hear Carmen talk about, the target indication as being for cases of late-stage melanoma where immunotherapy has failed. Melanoma will be the first, but we can see this therapy expanding quickly once proven there into any other of the significant solid cancer applications for which immunotherapy has failed. Thanks, Len. One more. And another click. So this is the team that we believe can take this across the line. We're very fortunate to have on our board level senior leadership that's relevant to what we're doing. Mark Levon in our chair is longtime head of Sanofi Pasteur. Walter Straps leads a biotech in Boston. Neil Fraser, longtime head of Medtronic Canada, and Jim McGann is a venture capitalist, 25 years of capital markets experience. So we've got the right mix, we've got a management team, and we have access to some important advisors, both in Canada and the US, on both the science side, the medical side, and the medical device development side. So with that, we'd like to thank you. We'd be happy to take any questions at this point. Len, you may be able to see there's a button you can press and to input your questions into the chat box. Len, you should be able to see those, and if you could share those with us.
on the chat. Oh yeah, here we go.
Just that chat box. Well, here's a question.
Here's one. Will large animal studies be required, or can you go right to human trials? And so based on the information we have now, we believe that rodent studies, and this would include rats as well as mouse studies, will be sufficient. And that's particularly true if we stick to intratumoral injection of the material. If we go to systemic injection, then the perceived risk is a little bit higher, and we probably would need, in that case, large animal studies. But if we go with intratumoral injection, we believe that the rodent studies will be sufficient.
And that's the specific feedback from the FDA to us. So, we feel really good about that and understanding that path. And there's another question asking about the significant
hurdles ahead and timeline.
And
so in terms of hurdles, there clearly are some hurdles.
And that Go ahead, Len. I think there's a lag on my speaking here.
Okay. Well, I'd say important hurdles are, of course, completing all of the preclinical safety studies. We have very good evidence so far that the injection of gold nanorods is very safe. The material is very safe, but we have to complete the formal GLP studies required by the FDA, of course. And those are significant studies. They take time, but we've got them mapped out, and we don't anticipate any huge issues there. I'd say transition of both the safety of the material and efficacy into humans for first-in-human studies is a major milestone, of course. And again, we're developing a plan for that as well.
Right, and I'll just speak to the timeline. Obviously, we are focused on getting first in human as quickly as possible and optimizing that path. That's one of the reasons we've chosen melanoma and pivoted from frankly, we came into this looking at colorectal, but for a variety of reasons, melanoma is going to offer us, we believe, a faster path to get into first in human with the goal of doing that in the next year. I want to move on here to, can you see the next question? I don't know, Carmen, you can see that, asking if endolymphatic injection was used.
Yeah, that's a great question. So technically, because of the subcutaneous intralesional injections we're giving, we are giving, there's an endolymphatic component to it. And so part of the analysis of the nodes is to actually, we're homogenizing nodes, and we're doing mass spec to see if we've actually delivered gold nanoparticles to the node. And of course, the question that we're looking at there is, if we have gold nanoparticle in the node and we have nodal disease, can we activate the nanoparticle in the node and generate immunity separate in the node from the tumor? So it's currently now it's being done kind of as a part of the construct of the experiment, but certainly it would be something we would be looking at for those deeper distant tumors.
And do you see that point on PDT might be a good adjuvant for cutaneous cancers?
I didn't see the question, but what was it?
Well, more of a statement, I think, a comment, which is welcome.
Oh, sure.
Okay. Photodynamic therapy might be a good adjuvant for cutaneous cancers.
Oh, absolutely. Oh, 100%. Yeah. There's no question. I mean, there's other cancers for sure will lend themselves nicely to this. Of course, the one that comes to mind for me is basal cells, which we struggle with, the rodents, the big ones. This would be a perfect next step. We have to choose one. And although basal cells have certainly a higher incidence, they're also dealt with more readily with just clear excision or even ablative therapies with cryo. But the melanomas that we see are the ones, of course, that create the problems because of their propensity to spread and travel along the dermal lymphatics, etc. So, yeah, there is, the field is as big as the imagination, and this is really tumor agnostic. One of the things I love about this particle is that as I suggested earlier, every tumor has abnormal antigen as a part of its domain. This is an opportunity to really see if we can expose antigen in those cold tumors.
That's it. Thanks, Carmen. Some commercial questions here around cash in the bank and financing going forward. we've been fortunate to benefit from some non-dilutive financing and run a very lean operation. We're now well into our efficacy study. We want to get the full efficacy study done. There are 2 more phases of it. Of course, Carmen mentioned colorectal model and a melanoma mouse model. Once that's done, we'll aim to hopefully have that data published and that study published. And on the back of that, certainly we're going to need financing to take us into phase 1 pivotal trials, but and through the preclinical work before that. But that's not a significant amount, as many large biotech companies, given what we have and the partners we've selected. Also, a question here on would there be a role for this kind of treatment in rheumatological conditions, cutaneous lupus, perhaps? You see that?
So that's an excellent question. As you know, if you're thinking of rheumatology, we've used gold historically in the treatment of rheumatological diseases like rheumatoid arthritis. The application as we use it, I don't really see the clear link, but that may be just my ignorance. The notion that we're heating the tumor, so if the process that's underway has some propensity to respond to stress, i.e. heat, and that can trigger an apoptotic cell death, that may be something. The issues in lupus, as you probably know, this is a sidebar discussion, but the IL-2 component, the low dose IL-2 may have some effect in lupus, as it tends to be a T regulatory cell phenomenon as well. So, but it's a great question. I don't think this is the direct application that we would be looking at in the near future.
So unless there are any other questions, looking forward, key for us in looking at milestones, we've got some significant, important milestones. We have a big impact on things, notably going to the next 2 models and seeing if these tremendous results are replicated in the melanoma model and the colorectal model. Getting this study published and then taking it to the various conferences, we think can represent a significant the commencement of the and an announcement around the path into first in human. There's one more question here. So the action of your therapy can be described as serum and vaccine impact?
Yeah, I love that question. That's one of my favorites, because we are actually looking at that in terms of a vaccine protocol. It's an individual vaccine protocol, much like an mRNA strategy, but with protein. And that is that we're by exposing the new antigen, the novel antigen, to the immune system, it creates an individual response to the tumor, and that's that systemic or abscopal response. It gets a little, so the answer is yes. It's a vaccination of sorts. You know, the term loosely applied in cancer, it can be problematic, the notion of vaccine, in that it's unlikely that I could take the serum from a treated patient and treat another patient with it, unless by coincidence that antigen is the antigen. They share commonality. Now, in time, if again, a lovely discussion, over time with banked tissue, we could create an antigen response, but that's definitely a for the future, goal.
But it's
it's definitely something that keeps you
the doors open to.
Yeah, absolutely.
Yeah. This part of the study is opening the door to a great deal of thought, and we'll look to sharing it with other folks who could become partners for whom it might. Carmen, you see that IL-2 question?
Yeah, again, these are great questions. Absolutely. If you're looking for toxic soup, you nailed it. You can definitely drive it, and we could look at IL-2, and we could look at IL-2 and IL-15. These are the exciting. Yeah, I believe this is as big as the imagination will allow. And again, it's this exposure and presentation of new antigen that just makes this all so exciting. So the marriage with as many immune Yeah, again, you look at the PD-1 expression on the CD8 positive cells infiltrating the tumor, and that's a natural response, as we know that. But that PD-1 expression it happens within 24 hours, and that early expression is an indication that there is maybe a more competitive antigen there. But in that initial response, we can use a PD-1 inhibitor as well, intratumorally or systemically, and a systemic delivery, which is already, of course, in the clinical environment. Combined with this could create even more effectively that abscopal response that we're trying to achieve. So I think that's going to be a lot of interest from partners that want to see what we have to offer. I'm hopeful for that.
Excellent. And a final comment here saying a long time ago, similar tactic was used for treatment of infections.
Yeah. Yeah, that's, that, and yeah. It's probably worth revisiting, in fact, for some of these difficult infections, the ones that have been difficult to eradicate, the ones that are deep in bone, the ones in tissue that have been irradiated, those kind of things. This is all reasonable stuff to consider in the context of the hyperthermia.
Excellent. Well, look, I want to thank Dr. Giacomo for his time, and everyone for joining today. We look forward to coming back to you and sharing results when we have data from, importantly, both the histology associated with the first phase of this triple negative breast cancer mouse model, and also the melanoma and colorectal models from mice. Those data. Really interested to see to what extent the great results we have here have been replicated, and we'll look forward to answering any questions. In the meantime, feel free to reach out to us. Always keen to discuss. Many thanks for everyone who joined, and we'll speak again soon.